So even though some tastings like sugars are attractive and other tastings like bitter compounds are aversive, that animals, whether you're talking about flies, rabbits, monkeys, and humans, can learn to like foods they didn't like before. The advantage of studying this process in the fruit fly is that we have genetic approaches at our disposal and the ability to use really simple behavioral assays and electrophysiology in combination with the behavioral assays. And so it's really the ability to bring all these different techniques to bear that make the fruit fly a wonderful model organism to study this process. It turns out that camphor is not toxic to the flies. So initially, when they've never seen camphor before, naive flies, they've never seen camphor in their diet, they're exposed to it, they will avoid it, they don't like it. But if they don't have the option to eat more attractive foods, and camphor is in the diet a long time, which to a fly is just a couple of days, they will learn to accept it. And the reason that they seem to learn to accept camphor and not some other types of tastings like quinine, which is toxic to them, is that the camphor isn't toxic. What's different about this study is how your taste proclivity changes with time based on your diet. And that's something I think as people we can all relate to, that as you're exposed to a diet in a different environment, perhaps you're going to a different country that has a different cuisine than you're used to for over a matter of weeks or a few months, you might learn to like foods that you didn't like before. And that's what's different, is trying to understand this well-known phenomenon that at a mechanistic level isn't so well understood.